All right, second video. I obviously talked too much, so let me move quickly here. The replicated chromosome. So now we're going to take a look at that a little bit more. And here's where we're going to mention the sister chromatids. But I like looking at this model over here. This is the DNA molecule. And at some time when the cell is going through its cell cycle, when it's in interphase, that's like kind of like its regular old phase, it will duplicate its DNA. It will replicate its DNA. When it replicates its DNA, it's still all loosey-goosey. It's not until after it replicates and it gets ready to divide that it starts bunching up. And this is a bunched up chromosome here, okay? This is one side, this is the other side. They are identical to each other, and they're held together by a centromere region. Okay, I know a lot of times in the past we would just say, oh, these are the centromere, which happens to be like one little thing. Well, it's not like that at all. Okay, it's just a region, and we're going to look at that a little bit more closer. But here's the DNA, okay, and this would be a single chromosome. It duplicates, then it shrinks up. It really doesn't go DNA, shrink up, duplicate, and then do that. It really duplicates or replicates when it's all stretched out, and then it shrinks up. So the first moment you see it, it's already replicated and shrunken up. When that cell eventually divides, one single chromosome will go one way, one will go the other way, and then that DNA will uncoil again to become normal again. Okay? All right, so that should be good. All right, now here's an up close and personal look at the centromere region. Okay, and those two sister chromatids. So I like this diagram because you see one and you see the other, and you see this centromeric region. It's really like a region that's made up of quite a few things. It's made up of these two proteins called kinetochores. Okay, and they're actually facing opposite one another. Then these spindle fibers, which are made up of microtubules, are in there, and they're kind of like plugged into these things. Okay, what's going to happen, and you're going to see it in the next slide, is those microtubules are going to be able to pull those chromosomes apart. But in the meantime, there's a little bit more. There's also protein holding those sister chromosomes together that are called sister chromatids together. That's called cohesin. So this replicated chromosome is still like one. Okay, even though it's like two halves, it's still like connected and all, and it's still like one. And that's why we call it one chromosome, even though we know it's going to be two chromosomes. Okay, and the microtubules are now plugged into that kinetochore, where those are in the centromeric region. So it's not really a centromere, so to speak. There's two proteins that the microtubules are plugged into. All right, now I know we're not looking at it yet, but mitosis. Let's go over those stages of mitosis. And in our materials, I gave you a picture of my notes from first year bio, okay, with my labels of like all the phases of mitosis, okay? And it's this little diagram here, okay, Mrs. Horner's pictures, okay? We have interphase. Notice I put a line here because interphase is really not a phase of mitosis, but prophase, metaphase, anaphase, and telophase are. Look through these again. Remember, prophase and telophase happen to be the busiest of the phases, just regarding the interphase, because you see the most going on then. You see the chromatin is becoming replicated chromosomes by coiling. The nuclear membrane disappears. Nucleolus disappears. The spindle appears. And if it's an animal cell, the aster appears and the centrioles split and move to the poles, okay? Ooh, pardon me with that. That's what's all going on in here. And we can see some of the diagrams here. In a plant cell, we don't have any asters and we don't have any centrioles. So we're just seeing all these other things happening. Metaphase is easy. I always say meta, middle. They're lined up in the middle. That's it. Okay. Anaphase, I think, is easy too. Ana, think the A's are separated by the N's. That's what happens there. The single chromosomes are now separating. They were replicated chromosomes here, like those X's basically, and now they're going to be single chromosomes once they split. Okay. And then finally, telophase. I always say telophase. Don't memorize all this stuff. And besides, you wouldn't be quizzed on that. But prophase and telophase are opposites. So whatever happened in prophase, the opposite is going to happen in telophase. Okay, but also in telophase, how do we split the two cells? That's that cytokinesis. Remember that word, cytokinesis. How do we split up the cytoplasm and all the organelles? Well, in a plant, you get this cell plate that forms, and we'll look at that in more detail. But in an animal, you get the cleavage for it. And again, we'll look at that in more detail. So going back to the PowerPoint, okay, there's our stages. And notice I threw in this pro-metaphase. This is because a lot of times scientists anymore, they're saying this prophase is so long. There's almost like a prophase and then a pro-metaphase and then metaphase. Not a big deal. Just remember prophase, pro-metaphase, same thing for me. And then cytokinesis usually happens during telophase, but some cells it happens afterwards. Here are the pictures that are in your book. Okay. They are all animal cell pictures. Okay. Again, it's just another look. Don't forget daughter cells. Okay. And you can see the cleavage for, that's how we know it's an animal cell. Okay. And now we're going to look at those spindles, pulling those chromosomes apart. Let's look at that in more detail here. Okay. So the more detail that we want to focus on is really that kinetochore. That's this whole part here. Okay. And it's enlarged. This didn't print out so well, but you can see the two sister chromosomes and the kinetochore is like right there and there. And you see that these microtubules are plugged into it. The ones that are plugged into it are called kinetochore microtubules. Okay, kinetochore microtubules. Those are the ones that are plugged in. So this right here, this microtubule is a kinetochore microtubule because it's plugged into the kinetochore. But there's other microtubules, like if you look at these, they call them non-kinetochores microtubules, okay, that just don't plug into the kinetochores. They kind of overlap. You're going to see that these non-kinetochores are going to grow longer. Let me write that down there. They grow longer. But the kinetochore fibers, the kinetochore microtubules, they're going to grow shorter. I guess I shouldn't say grow. Let's say go shorter. Go shorter. Okay, now how does that happen? Here's one of the kinetochores, and it's attached or plugged into that kinetochore microtubules is plugged into the kinetochore. Then we have these little motor proteins. Those are the little guys that I say, like when they get hit with ATP, they kind of like walk. They get boom. Every time a phosphate breaks off, boom, and it makes them walk, boom, and it makes them walk, boom, and it makes them walk. And as they walk this like tightrope of microtubules, they pull that chromosome apart. And that's what's happening during anaphase. But what happens to microtubule in the way? It kind of gets chopped off, choppy. All the little ends get chopped off into these tubulin subunits. That's with the kinetochore fibers, the kinetochore microtubules. But with the non-kinetochores, they're building tubulin units. So they're overlapped at first, but they start growing and growing. If they kept growing and growing and they didn't grow, but if the cell kept pulling apart, they wouldn't be overlapped after a while. So they actually grow and they always stay overlapped because they keep growing where the overlapping happens as the cell pulls apart. So the non-kinetochores help the cell like actually to expand wide, while the kinetochores actually shorten by pulling the chromosomes apart. Okay, so that's like a little different. You don't usually talk about that, but there's a really good video, and here's the little link to it. So if you go onto this PowerPoint, click that link and watch the video on how the spindle fibers are pulled apart. Okay, now that's important for metaphase to anaphase, pulling those apart. But the other thing that's important to look at, and remember, we always say AP Bio is just regular bio, but like in more detail. What's the more detail about cytokinesis? I know you're looking at that and you're saying it looks like a rear end or something, right? But what it's showing is it's showing the cleavage furrow in an animal cell. Now, how does that happen? I always say it's like, imagine you had a belt around your waist and somebody pulled it so tight that it pinched you in half, which would absolutely be god awful. But that's what happens in animal cells during cytokinesis, during that last phase, that telophase, when the cell has separated the chromosomes. It forms this band of microfilaments, not tubules, filaments this time, that kind of like just simply contract around it. Okay, and when it contracts around it, eventually pinches them off. That's how animal cells do it. But how do plant cells do cytokinesis? These vesicles, remember vesicles? You know how the DNA makes protein, which makes or DNA makes RNA, which makes protein. Well, the little protein then goes down the ER, and then vesicles pinch off. All these vesicles kind of form in the middle, and they just deposit a bunch of protein there. Okay, but they don't just deposit protein, they also deposit cellulose. Okay, and that's what's interesting in those vesicles. It's not just filled with protein, it's also filled with cellulose. They eventually build up, and if my pen worked, it would kind of build up, and they call this the cell plate. Sometimes they call it the cellulose cell plate because, yes, it doesn't just have protein in it, but it also has cellulose. Cellulose, let me write that. Cellulose, which is aka fiber. Okay, remember that's what the cell walls are made up of, cellulose and protein. That's what all comprises in those vesicles. Vesicles. Okay, they have cellulose and protein in them. And that all together will eventually cause what we call this cell plate to form. See how it does this? Then eventually cuts off the two cells. And that's just new cell wall like forming in between. The cell plate is basically a new cell wall. Okay, so when it came to mitosis, and let's go back to like Minot diagrams, okay, we went into a little more detail on the separation here, and we went into a little more detail of like how cytokinesis happens in both of them, okay? And so that's like kind of like your AP Bio, regular bio on steroids, okay? Now, let's talk about asexual reproduction. Asexual reproduction is when there's only one parent involved. The offspring are going to be genetically identical. This happens definitely in unicellular organisms. So here we have a picture of a bacteria cell, an E. coli cell. And they don't have strands of DNA. They just have one loop of DNA. It's all tangled around. So they too will have to replicate their DNA, but it's kind of interesting. They have a point of replication that happens, and there's like a duplicate that happens, and then that duplicate seems to happen until two like loops are pulled apart. Okay, so we have that happens, and then just simply the cell divides. So we don't see that prophase, metaphase, anaphase, telophase. We really only see like the replication happen, and then we see the cytokinesis happening. Okay, so we don't say that it's like thing. And one-celled organisms would use simple cell division like mitosis, called binary fission, to simply reproduce. But both new cells are identical to the parent cell. Now, where else is asexual reproduction used? This is going to talk about the evolution, first. So, you know how we all started off prokaryotic cells, right? Then eventually the nucleus developed. But how did cell division develop? Well, those microtubules that pull the chromosomes apart, originally they used to pierce. And you can still see this in dinoflagellates. They're a type of protist, a unicellular protist, that's kind of led to the evolution of animals. Those and plants too. Nope, they weren't the plants. Diatoms were the plants. Led to the plants and the fungi. But dinoflagellates led to the animals. Originally, the microtubules kind of pierced through. Okay, but we know in eukaryotes, the microtubules, what happens is the nuclear membrane disappears. So it all just happens here. So we get the nuclear membrane out of the way nowadays, but it used to stay intact, and the microtubules would just have to separate. Now, in the case of diatoms and yeast or plants, they were the originators of the plants, they formed their microtubules within the nuclear membrane. Okay, the nuclear membrane, again, stayed intact and all. But nowadays, we know most eukaryotes dissolve their nuclear membrane, and most of that nuclear membrane is used to like form the micro tubules, the kinetochores and non-kinetochores. Okay, so that's a little bit about evolution. And let me see if I can talk about the last thing before I run out of time on this. We have budding, which is a type of asexual. They would go through mitosis, where it would just grow, boom, eventually fall off. Regeneration or fragmentation, that's what's happening like with a salamander's tail. Propagation, vegetative propagation, I like to talk about is like strawberry plants when you see these little shoots come off. So those are just some other types of asexual reproduction. And I'm going to end this there.